pieces I saw this afternoon looked excellent, and I certainly was able to follow in the exon skipping area. And we saw the 79 sort of brick-looking structures. I get it with the right and left chevrons, or male and female, if you will, that you can sort of glue those bricks together if they fit properly. Or any two that have a flat end, if you can describe it as that, could be glued together. Are there patients whose deletion is between every one of the 79 exons? I know we talked about there being people at 50, 51, and you're able to bridge somewhat.
Right.
And also working on 45. It strikes me that, and again, I'm way out of my element here, that a lot of these exons don't appear to do anything. And when you talked about knocking them out, I took a look at the one slide, and it appeared to me that from 32 to 36, I stand to be corrected here, they didn't appear to do anything. What happens if you were to knock those out? Does it make the genes smaller at all? Or do the patients who have deletions still have a deletion exactly where they are, regardless if you have 50 exons or 79? I know it's a long, kind of crazy question, but I'm just curious about that.
Yeah, that's a great question, actually. There are many studies, impressive studies on BMD patients, and originally from 1990s. Is it on?
Yes, yeah, it's fine.
So from 1990s, 1980s, many researchers work on phenotype, symptoms, and BMD patients' molecular diagnosis, and it's quite striking because the length of truncated dystrophin is not really relevant or associated with the severity of the disease. So sometimes just 1/3 of the dystrophin, quite small dystrophin, it's called mini dystrophin, leads to very mild symptoms, and that led to microdystrophin research. Now Sarepta is working on clinical trials. So basically, microdystrophin studies are inspired by very mild Becker muscular dystrophy patients. They have very small dystrophin. So dystrophin is a very interesting gene because it's one of the largest human genes, but also the size of intron is strikingly different among the different regions. So about 50% of patients have mutation between exon 45 to 55, but other regions, for example, C-terminal regions, very few patients have deletion there. So there are hotspots in the exon 45 to 55 area, and also hotspot in N-terminal area. So many patients have mutations there, but other regions, not many patients there. So it's quite interesting, and eventually, hopefully, we can develop antisense oligos targeting each single exon from exon 1 to 79, and we can use combination of cocktail, so that hopefully we can develop therapeutics for 80 to 90% of DMD patients. So that's our ultimate goal of exon skipping.
I'd like to add on to that, please. Oh, look at that. So your question addresses a number of areas, and that was an excellent chunk of information. When it comes to deletions, now we know in the Duchenne population, 2/3 of the mutations are due to deletions, duplications, or point mutations, and 2/3 are genetically inherited, 1/3 are spontaneous, and then there is a portion. It's like another 2/3 approximately that are due to deletions, but then you also have a portion that are due to duplications and then point mutations. So First, you have 3 different types of mutations that can occur. Now, when you're we just look at deletions, yes, exons 45 through 55 are a hotspot for deletions and mutations. Which why if an antisense oligonucleotide that can address 45 all the way through 55 is developed, that is going to address the vast majority of deletion mutations. But when you have your various deletions, the dystrophin or microdystrophin or truncated dystrophin that is produced, if it is produced, can vary. Now remember, with some deletions, the in-frame deletions, you can still get a read-through and you can get some dystrophin produced. When you have an out-of-frame deletion, typically you won't have dystrophin that is produced. Even within those deletions, there are varying what we call phenotypes. It's the severity of the disease. So an individual with a deletion between, and I'm just throwing out random numbers here, 47 through 50 may have what we would consider a severe phenotype, where the individual loses the ability to walk early, loses the ability to use their upper arms early. Or you have another individual with deletions at another point in the Duchenne gene where they have a very mild phenotype. Perhaps they're ambulant into their teenage years, 14 or 15. So there's a great deal of variability just within the Duchenne gene and the exons themselves. And and I didn't even get into introns. So that's a whole nother talk in and of itself.
Other questions?
I have a
Yes.
question.
Yeah.
Just about exon skipping versus the CRISPR, because we're dealing with virus into the system, those folks who do partake in exon skipping would not be eligible for CRISPR then, or?
No, not necessarily, because exon skipping, when we're looking at PMOs and PPMOs, a PMO has a half-life of about, 14 hours actually? And it's completely excreted from the body in about eight weeks. Now PPMO, that only has to be dosed of once a month. And see, those are temporary. The body metabolizes them, the body excretes them. They're done, they're gone. So if there was a washout period following a PMO or a PPMO, yes, then the individual would be eligible for gene therapy, such as with microdystrophin, or once we get CRISPR into the human models, yes, they would be a candidate for CRISPR, because exon skipping, as we currently have it with PMOs and PPMOs, does not permanently alter the genetic material.
Yeah, so,
Oh.
thanks. So, in terms of morpholinos, PPMOs, and CRISPR, I think there's not much problem. I think more problem is, microdystrophin gene therapy and genome editing, because they both involve AAV-mediated delivery of genes. So, once you have injection with every vector, you may develop antibody immune system against every vector. So another injection with AAV might be problematic. So that's I think only concern I can come up with.
Or with the same AAV.
Yeah, that's what I mean. So there is a potential for, like you said, it could be problematic if you're dealing with the same.
It's possible. One of the biggest questions regarding current gene therapy, and that including SMA, is durability of response. How long is it going to last? And what do we do if the response doesn't last beyond, I'm just throwing out numbers, 10 years, and we have to redose? Do we have to use a different vector? Do we have to immunosuppress before we can administer? And these are questions that only time will tell. And right now, we simply don't have answers for them yet.
A second part of the question about the CRISPR, I know it's in preclinical, and I know you guys don't have a crystal ball, but can you forecast how long you think it's going to take? Any prediction?
I don't know. It's very hard to tell.
Yeah.
So we, I think one big problem in Canada is that it's hard to find industrial partner. There are not many pharmaceutical companies, big pharmaceutical companies in Canada, and we are currently discussing possibility to launch our own company with our collaborators and to move things forward faster. And so right now we have no, I don't have clear timeline yet, but hopefully we can have more clear timeline in the near future.
And we do have a partnership with Duke University. And this program very much so is in preclinical, and really all that I have heard is it looks good, but what that means as far as the timeline is unknown. So we'll keep staying tuned at the major conferences, AAN, MDA, and see what comes forth out of CRISPR. It's very promising, but it still is in the animal models.
Question for Nicholas. So obviously Health Canada is a big focus with regard to approvals, but as we know, CORD has to get involved and the provinces have to get involved and a whole bunch of different bodies. So can you touch on if there's any preliminary work with some of the other stakeholders?
Yep, no, for sure. Thank you. Yes, absolutely. And so one of the initiatives that Health Canada is currently working on is sort of co-review with organizations like CADIF, right? CADIF is our HDA organization. So typically what happens when a drug is approved by Health Canada, then it goes to CADIF to make a decision as to whether or not they're going to reimburse this. So when Health Canada approves, we may be able to get access to this treatment through perhaps our private insurers, but obviously these treatments are going to be quite expensive, and so it's not going to happen. And so we're going to have to wait until our province, until the provinces reimburse this. So CADIF is the agency that puts forth a recommendation in terms of whether or not a treatment should be reimbursed. And then it has to go to the individual provinces to then make a decision whether or not it's going to end up on their formulary or not. There's another organization called the Pan-Canadian Alliance, and so provinces can opt in to be part of a larger group that will make a decision on the reimbursement of this. And so they, whether or not that actually happens, and that takes a lot of time. So we have seen it take years in some cases, depending on how good the data is and what recommendation CADIF puts through. So to answer your question, what is happening right now is Health Canada is working closely with CADIF on a lot of new drug submissions. So they're sort of co-reviewing to try to shorten that amount of time between when a drug gets approved and when it gets reimbursed. This is relatively new, and so we still have to see what the outcomes of that, and we still don't know what the timelines of that will be. But that is one step further to shortening that time frame. With Duchenne, however, we don't have any treatments approved. I mean, even deflazacort, like prednisone, which is the standard of care, is not even approved for treatment in Canada. We all get that through the Special Access Program, right? So we have no bar on this. So our goal as a patient organization is, yes, we're engaging Health Canada, and we fully intend to engage CADIF and our HDA organizations at the same time. So that's our next plan, is to get them engaged. And when we have meetings and we invite our agencies to join, we're going to invite all of the relevant agencies to join, including CADIF as well. Because we want to ensure that we engage these agencies right up front from the very beginning, so they know we're coming and they know what our ask is going to be, that we can't sit by and wait for them to reimburse. The worst thing that can happen to all of us is that they approve drugs, but then no one can get access to it because we can't afford it. We don't want to be in that situation, but we may very well be in that situation. So we're going to do as much work as we can up front to try and tighten those timelines as much as we can, and that's our goal.
Any other questions from the panel? Yes.
I had a question for Jennifer. You're talking about the microdystrophin
Yes.
treatment. Is that agnostic to deletion?
Yes. The current clinical trials have a very specific deletion range. It's pretty broad. Oh goodness, off the top of my head, I think it's like 18 through 50 something. So it would be agnostic to deletion, because the way microdystrophin gene therapy works is when the transgene, when the vector is provided to the individual and the transgene enters the nucleus, it forms its own DNA. So it starts producing microdystrophin on its own. So it doesn't matter what the deletion is, it's going to produce microdystrophin. And so, of course, the hope is, as we continue through trials and we get more and more data, we expand the exon range and the age, that we discover that it's something that can be applicable to a broad population.
Thank you.
Sure.
I think the only concern is that newly introduced dystrophin can be immunogenic. Right?
Yes.
So, if patients don't have the specific region in dystrophin, and then transgene is produced, then that can be recognized by immune
Yeah.
system.
Yeah. And you're referring to revertant fibers?
Yeah, right.
So there are some muscle cells in the body where none of the muscle cells seem to be producing dystrophin, but there may be just a smattering, a handful here and there. And that being said, the body recognizes that dystrophin as being self. So if you were to introduce microdystrophin, there is a possibility that the body would just perceive that as foreign and then launch an immune response. So even with our study subjects, they are tested before they're ever administered for immunogenicity or immune response to the AAV vector and to dystrophin and microdystrophin to ensure that they wouldn't have an immune response, because, as you said, there are occasionally these fibers that do produce dystrophin on their own.
Right. Yeah, I mean, I asked specifically 8 through 13 are our son's deletions, and they're the 8 through 16, sorry, and they're obviously quite a bit more rare than where the majority of the focus is. So obviously we need to kind of look past where the focus is right now in terms of our silver bullet, if you will, in terms of really providing some better support. So that's where the question comes from. Thank you.
Sure.
Anything else? John?
Promise will be my last question. I just wanted to ask Ryan if you could tell us, what is the best example in your career that you've seen of advocacy working really well and really fast?
Well, it's primarily at the reimbursement stage. So let me make the distinction between the drug approval stage and the reimbursement stage. One of the things that Nicola touched upon was how groundbreaking the work that's being done today by all of you at the Health Canada level is, because that's true. Very few patient organizations that I've worked with and others have worked with over the years have done any advocacy to Health Canada, because everybody just said, well, there's no pathway, so we won't do it. And you all have said, we don't care that there's no pathway, we're going to do it anyway. So it is truly revolutionary and groundbreaking. So I have no wonderful examples in that space, because you all are kind of it. You're really leading the charge on that. So then you're down to sort of public reimbursement and how quickly you can get things done through public reimbursement. The challenge, John, is that there is this process that Nicola described. There is this step-by-step process that you have to go through. And that process is inherently built to take time. And I'll be even more pragmatic. It's inherently built to take time so payers don't have to pay. That's in part, this is my opinion, why it's built that way. So first you have to go through the health technology assessment process. Then you have to go through the Pan-Canadian pharmaceutical process. Then, even after all of that is done, you still have to go down to the provincial process, and that can take some time because each province is entitled to reimburse as a province. You know, we have 19 public drug plans in Canada. 19. Each one's different. Each one's independent. Each one does what they want of their own volition, sort of thing. So I don't have a wonderful example of anything that's really sort of sped its way through the process, John, because the process kind of is the process. But I do want to leave you with one thought, though, and I'm not answering your question, but I do want to leave you with this, and it's important. This process, this HTA process, this Pan-Canadian Pharmaceutical Alliance process, and all the rest of it, is not grounded in anything constitutionally or legally. It's made up. It's the provinces and the federal government having come together to decide we're going to create this. Understand that after a drug is approved by Health Canada, the next day, the province of Alberta has the legal right to reimburse it. They don't have to wait for any of that stuff that has been put in place. They're not going to reimburse it the next day because they rely on those processes to slow down what happens, because every day the government doesn't pay is a good day in any healthcare circumstance. That's just how our system works, unfortunately. So it's up to us to try to remember and to look for those pathways where we can expedite that process, even though we're told, no, you're supposed to jump through all of these hoops. But a lot of the work that we're now doing is saying we're not accepting those. We're not accepting that. There's no foundation to enforce that we must go through these hoops. So we want to go through something quicker. And in fact, that's how it was done at one time. 15 years ago, after a drug was approved by Health Canada, the provinces simply reimbursed them like that. It was very quick. That's not how it works anymore. They have created, in between those two steps, a process that now takes a year, year and a half, or 2 years that it adds to that process.
And if I can add to that too, it's why the work that we're doing is so important, to make sure that we are in touch with our political leaders in each of our provinces, right? That we make them aware of why we have a sense of urgency, why we can't wait 1 year or 2 years, right? Think about what 1 year or 2 years means in the life of your child, right? We need to be able to communicate that to our political leaders. And tools like the e-advocacy hub, for example, is a great way to be able to do that and to start meeting with your political leaders. It's almost like we're priming everybody for what's to come, because when those drugs do come here, and they will, and when they get approved, we want to be able to like flip the switch and turn it on, right? So we need to set all the soldiers in alignment to get ready to be able to do that, which is why a lot of the work of what we're doing right now is so critical and so important.
The back. Yeah.
I just had a quick question about that, about drug approvals and stuff, because we get, like you mentioned, deflazacort here on a special case. So we pay a minimal nominal fee for that. It's actually, I think, what the equivalent in US dollars is to exchange it through Mexico. I think that's where we get from. In the United States, it was approved at $5,000 a month. It's a corticosteroid. It's a cheap drug. It's ridiculous. So when they come here, if they do get approved, is that necessarily a good thing? Like, I'm really happy it hasn't been approved here. We still have access to it, but we're not paying that kind of money.
Yeah, so there's probably a reason why deflazacort hasn't come here, and I don't know if PTC wants to talk a little bit about what's going on there. I mean, for example, deflazacort is available in the U.S. And I believe it's something like $30,000 for that. I'd be quite surprised actually if we have that come through in Canada, only just financially, I think from a pharmaceutical standpoint. It's going to be really tough for them to sell a drug at a much higher price when we're already getting access to a product. I mean, we still pay for it out of pocket, but it's not, nowhere near that price. But you make a really good point, and I'd like to make another point too, is, you know, we have drugs like deflazacort that we are getting access to through special access program. And I think here we can all argue that the safety profile for a drug like deflazacort is quite substantial, right? And you think about the side effect profile of deflazacort. So I think, you know, we have other drugs that have been approved in other countries. They're obviously not a steroid, but the safety profiles are certainly much more favorable, and yet we're having trouble getting those drugs approved. But there are other drugs in the pipeline that are looking very promising, and for example, one of those is vamorolone, which Laura spoke about. I don't know if you want to talk about that, but I think that's, sorry, sitting right here next to us. But I mean, that's a very promising opportunity, I think, for Canada, and I'm going to let you talk about that.
Is this on? So it's so, I have to say
Okay.
it sounds so weird up here, you're not hearing your own voice. Yeah, I mean, I think, you know, we, as I mentioned during my talk, we have to continue to the completion process of that 004 study to confirm the results that we have. But I'm encouraged that it'll have a better safety profile and that we will be able to bring it to Canada at some point.
And did you want to
comment
on that? Sorry,
I'll take this real quick. Yeah, just to comment on the price of the drugs, specifically Emflaza in the U.S. As a rare disease patient as well, the cost of my drug is extremely expensive. I get that explanation of benefits from my insurance company every couple weeks, and when I see it, it's like just crazy expensive. At PTC, as I said, we are committed to the Duchenne community, and the majority of the patients that are on Emflaza in the U.S., they pay, it's less than $2. So I kind of breezed through one of the slides that talked about PTC cares and how to get access to the drug. And again, most of the patients pay under $2. Most of them are no out-of-pocket costs, and that's important for us at PTC, is to make sure that we are able to provide the drug to the patients. We acquired Emflaza actually through a different company, and again, it was mainly because of our commitment to the Duchenne community. We saw an opportunity to purchase this company so we could get the drug to 100% of the boys and girls, but mostly boys in the U.S. In the past, there was only very few patients that actually could afford Emflaza because there was no special patient assistance program, there was no bridge program, it was all out of pocket. So they were getting it from either Canada, India, Mexico, and again, when they're getting it from different countries like that, they don't know exactly what they're getting. When it is FDA approved, they know exactly what they're getting, and again, our commitment to the community was to purchase this drug so 100% of the boys that wanted to be on Emflaza could be on Emflaza. And again, just to reiterate, it's little to no out-of-pocket cost for those patients. So,
thank you, Brad. In the interest of time, and we do have an opportunity with the social to carry on the conversation with any of our panelists, family to family, researcher to researcher, whatever. And we have just about 15 more minutes of the program, and then we'll wrap this up. But I want to turn things back to our Executive Director, Perry Essler, if I can. And thank you, panel, very, very much. And you may be facing a few more questions later.